Hello, I'm Hannah Donnert with the Collaborative on Health and the Environment. Che enjoys bringing you the latest environmental health science through our partnership calls, webinars, science serves, publications, and social media. I would like to welcome everyone to the first webinar in our new partnership series, Generation Chemical, How Environmental Exposures Are Affecting Reproductive Health and Development. This webinar series is brought to you in partnership with the University of California, San Francisco Program on Reproductive Health and International Federation of Gynecological and Obstetrics, the Alliance of Nurses for Healthy Environments, the Endocrine Society, the International Federation of Fertility Societies, and UCSF's Environment Research and Translation for Health Center. We will leave time following the presentation for a brief Q&A session. You may type in your questions through the Q&A feature available on the menu bar at the top of your window at any point during the presentations. After the presentations, our moderator will read out questions for our speakers to respond to, and we will also have a panel discussion. We will get to as many comments and questions as we can during the Q&A period, and we'll follow up on unanswered questions later. For those of you who have called in on the phone, we have posted slides to accompany today's webinar on our website. You can download these by going to healthandenvironment.org. Please scroll to the bottom of the page and select today's webinar. On the web page is a link to the slides. Everyone in our webinar right now is muted with the exception of our moderator and our speakers. The chat feature is open to all of our attendees though, so we encourage you to feel free to talk via chat. This webinar is scheduled to last for 70 minutes and is being recorded for our call and webinar archive. With that, I'm pleased to introduce our moderator today, Dr. Tracey Woodruff. Dr. Woodruff is the director of the Allison S. Carlson Endowed Professor for the Program on Reproductive Health and the Environment. She is a professor in the UCSF Department of Obstetrics, Gynecology, and Reproductive Sciences and the Philip R. Lee Institute for Health Policy Studies. She's also the director of a newly awarded NIEHS Environmental Health Core Center grant, the Environment Research and Translation for Health Earth Center at UCSF. She's a recognized expert on environmental pollution, exposures, and impacts on health with a focus on pregnancy, infancy, and childhood, and her innovations in translating and communicating scientific findings for clinical and policy audiences. She's authored numerous scientific publications and book chapters and has been quoted widely in the press, including USA Today, the San Francisco Chronicle, and the New York Times. Before joining UCSF, Dr. Woodruff was a senior scientist and policy advisor for the US EPA's Office of Policy. She was appointed by the Governor of California in 2012 to the Science Advisory Board of the Developmental and Reproductive Toxicant DART Identification Committee. So with that, thank you for moderating today, Tracey, and I'll turn things over to you.
All right, well, thank you, Hannah. And I want to thank the Collaborative on Health and the Environment and Karen Wong, who's the director, who reached out to me a few months ago, maybe it was the summer, to say, you know, we have had a lot of science happening around exposures to environmental chemicals and their influences on reproductive and developmental health. And I know we also have Dr. Linda Judice on the phone, who's going to remark on the amazing trajectory that we've had in the science since the summit that we hosted with Che back in 2007. And it just seemed, given where we are in this moment in time, that it was an excellent time to stop and pause and review where we are on what we know about chemical exposures and their impacts on environmental health and their generational impacts on health. So we have collaborated to put together this series of webinars that will talk about various aspects of this problem and some of the solutions to addressing it. So we'll be talking about exposures to chemicals, how they affect reproductive health and fetal development. We'll also have be talking throughout the series about disproportionate impacts on communities of color and low-income communities, and we'll also have one webinar dedicated to this very important topic. We'll talk about the infertility crisis. We'll talk about preconception and prenatal exposures, and also how they affect male and female health as well as child health. So I hope that everyone can continue to join us for this webinar. We're really thrilled to have our clinical partners. I know Linda will probably mention this, but since 2007, we've done a tremendous work to engage with clinical partners because they're a very important part of our strategy for reaching patients and the public, and of course, advancing protective health policies. So I am going to now introduce the speakers for our kickoff session. We're really thrilled to have Dr. Shanna Swan, Dr. Germaine Buckley, and Dr. Jessie Buckley, who will talk, provide an overview for us. I'm going to introduce them briefly, but probably most of you know them because they're very famous in our field. Dr. Shanna Swan has worked for over 25 years on understanding endocrine disrupting chemical exposure threats and how they may impact how they do impact male reproductive health. And she has some very exciting slides to show us about male infertility. She's at Mount Sinai School of Medicine in the Department of Preventive Medicine, and she's worked with, I think what's really going to be fascinating about Shanna's work is her work that she's done with epidemiologists and toxicologists to really produce groundbreaking work on phthalates and male reproductive health, as well as evaluating human epidemiological studies. And then we'll have Dr. Germaine Buck-Lewis, who's a reproductive and perinatal epidemiologist, and she focuses on environmental exposures also to endocrine disrupting chemicals, as well as stress and other factors on reproduction and development. She's been in this field for over 30 years, and she has done really groundbreaking work on fecundity and fertility, and she's going to talk about that from some of the studies she's led. She's currently professor and dean of the College of Health and Human Services at George Mason University. And on a personal note, she's been very excited to talk about science today because, as you can imagine, people who are leaders at universities have a lot of other COVID-related responsibilities. So we're glad that she could take time to join us. And finally, I'm really excited to introduce Dr. Jessie Buckley. She's an assistant professor of environmental health and engineering and epidemiology at Johns Hopkins Bloomberg School of Public Health. And she has been doing some very exciting work on characterizing early life chemical exposures and their effects on child development using molecular epidemiology and advanced statistical approaches. She has an NIEHS Outstanding New Environmental Scientist awardee, and we are collaborating together on the NIH Environmental Influences on Child Health Outcome Studies. So I know she has some very exciting slides to share with you. So I introduced the speakers so that they can get right into it. But first, I want to turn it over to Dr. Linda Judice, who has been a really important leader in this field. And she's going to give some opening remarks for the series. Linda?
Thank you, Tracy, and welcome, everyone. Today is very special. This is the inaugural series for this program and truly a collaborative effort. It's now 13 years since our 2007 summit that we held at UCSF, and the field has exploded and the partnerships are increasing by the day. Today is a partnership essentially with PHRE, the International Federation of Obstetricians and Gynecologists, FIGO, the International Federation of Fertility Societies, IFFS, also the US Endocrine Society, and the Alliance of Nurses for Healthy Environments, and of course the UCSF program for reproductive health and the environment. So it's an amazing time. It's very timely for us to be talking about chemicals and also climate change and their effects on reproductive health and also development. So I want to thank Tracy for inviting me to say a few words. And without further ado, I think it's time for the program. So thank you so much.
Great. Now we'll turn it over to Shanna. Thank you.
Thank you, Tracy and Linda, and thank you for allowing me to kick off this very important program today and this very important series. So today I'm going to start our discussion by talking about men's reproductive health and the environment, and in particular, the question of declining sperm count, which is only the tip of the iceberg. For some reason, I can't change this slide. Okay, there we go. Thank you. So I've been tracing this problem, as Trace told you, for 25 years. But in 2017, I think we had a breakthrough. We had the publication of a study which actually went viral around the world. And we announced, and Newsweek and New York Times and others announced that this was a decline that was really baffling. What we reported was that sperm count had declined 52% in 38 years among men from Western countries. And we looked to see whether this was a continuing decline, whether it might be tapering off, and we saw absolutely no evidence that there was any tapering off. So this is an image of the cover of the Human Reproduction Update volume in which it was published, and the article. And this paper actually was, I think, 28th most cited paper in the year, and caused a big stir. And one of the things it did was to get a literary agent to come to me and say, would you write a book about this? And I'll come back to that at the end of this discussion. So this analysis, this meta-analysis, which we published in 2017, was a huge effort, the result of the work of a team of 8 people from 6 countries. And our goal was to conduct a systematic review and meta-analysis of sperm counts concentrations in the years 81 to 2013, which was the last completed year for our study. And here you have the picture of our team, and the leader, the first author, was Haggai Levine of Israel, now very active himself in COVID response. Our study was new in a lot of ways, but it was not new in showing a decline in sperm count. And here I've put up 3 images, 3 figures from 3 very different papers in terms of time, showing basically the same trend. So the first of these on the left is a study which was published in Denmark by Elizabeth Carlson in 1992, in which they concluded that there had been a, quote, genuine decline in sperm count of 1% per year. And this paper was widely criticized, attacked, and many people did reanalysis, and I did one as well. And what I'm showing you here is in 2000, I actually did another reanalysis, another analysis which went into other factors like geography. And though we accounted for all the potential biases and confounders we could think of, such as change in population, change in method of counting sperm, age of the men, obesity, smoking, and so on, we could not make this go away. And in fact, if you compare these 2 curves, you'll see that the overall trend is about the same. You'll also see that the green line in other countries is very small and quite flat, and that's because we really don't have information from other countries. All of these data pretty much are from Western countries. And in the third graph, which is on the right, you see the results of our recent 2017 study. And in that, we actually presented 4 lines. One was Western unselected men, and the other Western populations were the fertile men. So we had one finding on fertile men, one on unselected men, who many didn't know their own fertility. They tended to be young. And then we had the non-Western countries, again, with very little information. So what could be causing this? These declines are really rapid. They're unlikely to be genetic because they're so rapid. That means that they're environmental. And that environment, of course, does not mean chemical. It means many things. But we wondered about the role of testosterone because this is so critical to the development of the genetically typical male and female genitalia, which differentiate early in pregnancy. So we asked, was there something interrupting this differentiation that might lead eventually to these outcomes we're seeing. So we asked what could interfere with testosterone, because that is so critical for that differentiation at that time. And here I've shown you 3 pictures typifying phthalates, certain phthalates, and that was actually the class of chemicals that we honed in on originally because they are widespread, because they are potent anti-androgens, several of them, and they can decrease testosterone. So to look at those, we looked at animal data, and we could see that in this important paper from 2000, that the testosterone surge that's expected around day 18 to 21 in rodents, 19 to 21, 22, it's, not specific at the ends. If you give the mother diethyl hexyl phthalate, it absolutely wipes out that testosterone surge. So this is really important because this is the time when the differentiation of those gonads is taking place. So the experiment of giving the mother diethyl hexyl phthalate, one of the more potent anti-androgenic phthalates, at this critical window resulted in something which in 2000 was termed the phthalate syndrome. And this is a syndrome in males, which consists of a smaller penis, a malformed penis, undescended testicles, and then the shortening of a measure which hasn't been widely used in, and certainly in human studies until we started using it, which was a shortened anogenital distance. So what is that? So it's a marker of the distance from the anus to the genitals. Here's a picture of a rat anogenital distance in a male and a female. You can see that the male is much longer. In fact, it's 50 to 100% longer in males than females, and it reflects how much androgen the rat was exposed to during fetal development. Now, this has been known for 100 years approximately, and it was used for looking at reproductive toxicity of chemicals by various regulatory agencies, but it was not used in humans for human toxicity until we applied it. So what we decided to do was pose a hypothesis that prenatal phthalate levels in the moms of boys, but not girls, would be linked to their genital development, an alteration of their genital development. And so to look at that, we looked at the phthalate levels in mid-pregnancy urine samples in a pregnancy cohort that we had ongoing. And we then examined the children using a special examination that would look into these measures, including the anogenital distance. And what we found was indeed a phthalate syndrome in male infants in the 2 pregnancy cohorts. And by the way, no effect on females. So the boys born to mother with higher phthalate concentrations did have more undescended testicles, less testicular descent, a smaller penis, a smaller AGD, a smaller scrotal size. So we did find the counterpart of the phthalate syndrome, and we actually replicated that in a whole different cohort 10 years later and found very similar results. So, so what? So why do we care about a shorter AGD? And that's a good question. The boys don't look malformed. You wouldn't know to look at them that they were affected. But it turns out that AGD, when it's shorter, is associated with male genital defects, hypospadias, and cryptorchidism. And these 2 studies are examples of studies that show that adult men's AGD is related to their function, their reproductive function, their fertility, their sperm count, which is of course not something you can look at in the infants. So there's a gap here. We haven't followed our measured infants until they're producing sperm. In animals, there does seem to be continuity that AGD is, quote, forever. But if that is the case, then a short AGD at birth will be predicting poorer sperm count and more infertility in the adults. So the bottom line here is that sperm count is declining 1% per year in Western countries. Phthalate exposure in early pregnancy causes the phthalate syndrome in animals and humans. Men with low sperm count have shorter AGD. So there is a link between this data on AGD and our 2017 publication of decreased sperm count because exposures have been increasing. And it's not just phthalates. There are many things that can alter hormones, that can alter androgens, that can alter these measures of male reproductive function. But the data are limited, and I want to just put out a warning on these limitations, which was something that I'm happy to tell you the organizers asked for a slide on equity in these data. And these are basically data of white non-Hispanic men. They are data from Western countries. And even though there are many endocrine disrupting chemicals that are higher in non-whites, they're not well studied. So what we really need is studies of male reproductive function in minority and non-Western countries. And we need this badly and now. So then just an action slide. There are some changes that I think have to be made if we're going to address these problems. We have no monitoring currently of reproductive health endpoints as we have for cancer surveillance. We need a monitoring of reproductive health endpoints. We need to advance the creation of safe alternatives to the hazardous chemicals that are causing these changes. We need to develop testing protocols that can protect human and environmental health. Our current testing protocols do not do that. And we need to educate our regulators, healthcare professionals, and consumers about the risks of EDC. Finally, I'd like to tell you that this discussion and many other topics are covered in the book that I wrote with Stacy Colino that's coming out on February 23rd. And you can ask me questions at this info@shannaswan.com. And you can actually pre-order the book on a number of websites, including Amazon. So thank you very much for your time, and I look forward to hearing the other presentations.
Thank you so much, Shanna. While we're waiting for our next speaker to pull up our slides, we would like to remind you that you can put in questions in the Q&A box on Zoom, or feel free to chat and let us know what you're thinking. And it looks like Germaine is ready. Go ahead. Germaine, I think you're on mute. Oh,
there we go.
Great.
All set? Can you see it okay?
Yes, it's great. Thank you.
All right. Thank you. I said good afternoon, everyone. Then I realized on the West Coast, you're not quite at afternoon time. So good morning there too. And just a quick shout out to Che and all of your partners for thinking about this great series that's going to happen. And I look forward to hearing over the next several sessions from leading authorities in the field. So hello. So I don't know what I was thinking when I agreed to the title, Endocrine Disrupting Chemicals and Female Fecundity, Implications for Health Across a Lifetime or Lifespan, and how I would approach that and then do it in 15 minutes. So in thinking about that, to try to organize the talk, I've presented the slides in the context of why this whole topic is important, a little sense of what we know, and also next steps. So let me just say a few words about endocrine disrupting chemicals. I'm interested in these exposures because most of us have exposures, maybe in varying degrees, but we're talking about very prevalent types of exposure and not a rare exposure, as might be the case with other types of environmental exposures. And their ability to really impact hormonal milieu begins to tell me that the multitude of potential effects could be quite enormous. the good news is they're easily measurable, and increasingly we're reminded and encouraged to think about chemical exposure in the context of other important aspects of lifestyle, such as stress and perhaps diet. And still, the timing of exposures during sensitive or critical windows of human development, we don't always have that luxury of getting it right, or perhaps even knowing when it is. And the challenges of understanding the multitude of environmental exposures that may be impacting human reproduction. So just to clarify, for fecundity, I use this term in the context of more of a demographic definition, and that is the biologic capacity of men and women for reproduction, irrespective of pregnancy intentions. And I'm going to give you I guess the takeaway message now, and that is, I am convinced over the 30 years that I've been working in this field that we now have ample evidence to suggest that EDCs and other environmental influences impact fecundity, which probably is in the pathway to health across the lifespan, and potentially multi or trans generations. And that part is a bit scary for me. So I've been trying to make the link between fecundity and health, and I'm going to talk a little bit more about that. I believe we have considerable amount of data for various classes of endocrine disrupting chemicals suggesting associations with impaired fecundity and other health states. I don't think we have found a vitamin or any evidence to suggest that these chemicals actually are pro-health in any way. And the absence of maybe effects, but also in the context of no vitamin, needs to be considered. We still don't have a population biomarker of many fecundity outcomes, and I'm humbly reminded for couple-dependent outcomes like pregnancy, we're talking about the couple's fecundity. So in terms of health across the lifespan, all this has implication why it's important, because it allows us to be thinking about primary and secondary prevention strategies to keep people healthy, to make health visible. And we know, this has been a long-standing premise of the World Health Organization and many, many other entities, that health is a continuum and it's not merely the absence of disease. And that is something that I try to keep my own work grounded in. We certainly need to get the causal inference and the causal pathways right, so we can say things like, do fecundity impairments cause later onset diseases? So this is the overarching context for today. So I just wanted to put out the definition of endocrine disrupting chemicals, and it's one by Zoller et al. That I really do like, because it's simple and it's widespread in the sense that endocrine disrupting chemicals can be an exogenous chemical or mixture of chemicals that interfere with any aspect of hormone action. That immediately tells me it's a complicated question, but it also gives some biologic legitimacy for thinking that we should be looking for many different health outcomes and not just one, depending on how hormonal milieu might be affected. And these photos are just examples of how you might be exposed to some, but certainly not all. And I try to pick ones that resonate even for people who think they live a healthy lifestyle, just to remind people that we do have exposures as a part of living in contemporary society. So other speakers are going to be speaking to you about sensitive windows, timing of exposures during sensitive windows. And rather than put up that classic embryology slide about sensitive windows, mostly for structural defects, I think it's important to emphasize that sensitive windows can be across the continuum of age and generations. So I like this slide, even though it doesn't show very much in terms of conception or preconception sensitive windows, to remind us that we're talking about a continuum of sensitive windows as well. And I'm very humbled by the fact for female fecundity, at least, at the population level, we really do not have strong biomarkers of things such as ovulation, as shown here on the left, or of conception. So I think we have to be humbled that we use a lot of proxy measures when we're thinking about human fecundity. So this slide is my attempt to try to summarize some of the literature looking at various types of endocrine disrupting chemicals, the classes shown here on the left, and those that have been linked to diminished fecundability, meaning a decreased probability of pregnancy per menstrual cycle, resulting in a longer time to pregnancy. So these are just some of the authors who have published showing linking endocrine disrupting chemicals to diminished fecundability. And on average, we see about somewhere between a 10 to 20% reduction each cycle in the probability of getting pregnant. And I have a few of the papers in purple underscoring that couples were looked at, not just women. And if we're thinking about time to pregnancy and we recognize that it's a couple dependent outcome, maybe we should be looking at males. And the reason I think that males are important have really resonated with me in the results of some of our work from the LIFE study that was done while I was at NICHD, along with other investigators across the country. And let me just orient you to the slide. This is looking at, we looked at over 123 different endocrine disrupting chemicals, and these are the results, the significant reductions in fecundability or a longer time to pregnancy for couples. And when we looked at her, we had a few hits, 4 chemicals suggesting about a 20% reduction based on her chemical concentration. But if we looked at her male partner, and these are results controlling for the other partner, we saw many more different chemicals being associated with the couple's time to pregnancy. We found one vitamin here, PCB 101, congener 101. And then when we looked at the non-persistent, the short-lived chemicals like phthalates that Shanna was talking about, and sunscreens and antimicrobials, only one signal on the female side, but several on the male side. And it humbled us as researchers to say, if we only looked at females, our conclusions would have been a little bit different had we not looked at the couple. So I've been on a little bit of an environmental repro campaign to underscore how important it is to try to look at couples as a unit of analysis when looking at couple-dependent outcomes. And I know sometimes that can be a sell through study sections, but I'm hearing from my colleagues that over time thinking is changing, because it seems, at least in the LIFE study, he was driving most of the reduction in fecundability, resulting in the couple's longer time to conception rather than her. So this is the only study that I am aware of, and if there are others, I'd love to hear about it in the chat or send me an email. I like this study because this study was following up women who were eating fish from Lake Michigan with considerable PCB exposure. So a few decades back, those women had their PCB concentrations measured. And then the investigators followed up their daughters who were exposed in utero to the mom. And what they found a generation later, that girls born to mothers with higher PCB concentrations also had a reduction in fecundity or a longer time to pregnancy. And here, the most highest exposed girls, about a 60% reduction in probability of conception relative to the lowest exposed. And this was, they asked the women about all the times that she had unprotected intercourse. And they also did a restricted analysis looking just at the times when the woman said she was actively trying to become pregnant. We saw an even greater reduction based. So this is suggesting potential generational influence on fecundability based on mom's exposure. So now moving from or staying with fecundity, and I think of endometriosis and all gynecologic disorders as female fecundity impairments, I summarize some of the literature, or most of the literature that's available, on whether or not these chemicals influence endometriosis, especially concern for estrogenic chemicals, in recognition that endometriosis is an estrogen-dependent disease. And so these are just some of the examples that are out there, but there was also a very recent meta-analysis done for endometriosis, looking at the published literature, finding 3 classes of EDCs being associated with an increased odds of being diagnosed with endometriosis based on the available literature to date. So in thinking about fecundity-related impairments like PCOS and endometriosis here, and increasing evidence, in my view, that they're both associated with gravid disorders during pregnancy, and then increasingly being linked to a whole host of chronic diseases later in life. This suggests that maybe there is a shared exposure or a common pathway. I still keep thinking about, what does that mean for the offspring? And we do not have much data where generations have been followed with women born to some of these conditions. There are some, but not a great deal. And increasingly, I like this quote from Marcel Cedars about reproduction maybe being the 6th vital sign for clinicians to consider. So collectively, we tried to put this all together, working with many colleagues and some fellows a number of years ago, basically wondering why there wasn't ovarian dysgenesis syndrome being discussed, much like the testicular dysgenesis syndrome that was everywhere. It seemed to be in all the clinical journals, environmental. So I tried to convince my colleague, Neil Skakabak, to think about doing this with me. And he said he had enough work trying to convince scientists and clinicians to think about TDS. So in thinking that the gametes aren't really all that different, maybe there was something there. So this was just an attempt to parallel the TDS paradigm to say, maybe it's possible that gene-environment interaction or environment alone perhaps can impact ovarian development in utero, and whether it's through follicular genesis or steroid signaling, we can see a multitude of potential health outcomes. And this was directly in response to all the reviewer comments that I got saying it's unlikely that a single environmental chemical could produce so many different adverse outcomes. So this is something that it's helped us to think through how we might go about answering these questions. So in terms of thinking a little bit about policy and the so what of all of this, if you're of the mindset that fecundity impairments, gravid health disorders, and adult diseases, chronic diseases are important for the health at the population level, if not the globe, it calls into question at what point do we work in support of trying to protect health. The action, the MAID action that Shanna talked about really resonates here. Because we don't really have very good surveillance data. And so we wait many, many years until we can really be able to assess some of these changes over time. And even in terms of the clinical side, the woman before you, if she's there for a pregnancy or trying to get pregnant or something, is going to go on clinically to see many other types of clinical specialties as she moves out of those pregnancy years. And how do we begin to start thinking about health over the lifespan from a clinical perspective that may be more interconnected? So we all know that pregnancy is a so-called ultimate stress test there is, but it may be that reproductive health is really the quote unquote biomarker of population health. And by close attention to it, we may better be able to plan for health across the lifespan. So I'm just put this slide in. I'm not going to go through it, but I do feel it's important for the audience to know that there are concerted efforts out there and very good publications looking at causality, chemicals that cause, whether it's cryptorchid testes or testicular cancers, more ART treatments. The evidence suggests that there are very strong associations with potential causal impact. And these are very costly for society and costly in terms of those affected with these conditions. So my action call certainly is to follow up Shanna's, because I did see her slides earlier and I really did like her MAID. I would just put a little tag underneath the MAID and maybe say together, because I do know that these early clinical calls that came out a number of years ago already helped legitimize, in my view, the epidemiologic work that was coming out suggesting that environmental chemicals were impacting human reproduction and development. So I'm really very appreciative of these early works. So on that note, I just want you to know that we're working very hard to make health visible in our college to ensure people have the chance of having health across the lifetime. And I'm very appreciative of Che and all of the supporting partners for this opportunity and for the incredible series that will be forthcoming. So thank you.
Thank you very much, Germaine, for your great presentation and thoughtfulness on the topic. And we're waiting for our next speaker to pull up her slides. Jessie will be doing that momentarily, and we'll go ahead and pass it off to you, Jessie.
Great. Thank you so much. Again, I want to echo the other speakers and say I think this is a fabulous webinar series, and I really want to thank the partners and Che and Tracey for moderating. So I'm going to be talking about chemical exposures during critical developmental periods, and in particular, how we can address some of these unanswered questions. As the other speakers have noted, there are a lot of unanswered questions, and how the NIH environmental influences on child health outcomes, or ECHO program, and to start answering some of these questions. So this is the figure that Germaine alluded to earlier, showing how exposures during different periods during development can affect different body systems based on the timing of exposure during gestation. Early life is highly susceptible to chemical exposures due to the rapid development of the fetus from a single cell to a walking individual in about 2 years. When mothers are exposed to chemicals during pregnancy, the chemicals can cross the placenta and expose the developing fetus. And so exposures during these critical windows can cause permanent changes to the structure and function of body systems. And as Germaine mentioned, increased susceptibility doesn't stop at birth, especially given the continued maturation of the body, unique behavioral patterns of children, and higher exposures per body weight. And so there are a lot of studies out there that have shown that environmental chemical exposures during early life can impact child health and development throughout the lifespan. And again, these gestational exposures to the mother can also expose the fetus and the germ cells of her grandchildren. And so there's this real potential for transgenerational propagation of health disparities. So when a woman enters pregnancy, she may have low exposures and low vulnerability, or she may have high exposures and high vulnerability. And this vulnerability can be environmental exposures, and it can also be social factors such as poverty, racism, worse access to healthcare, psychosocial stressors, and other factors. And these can all interact with the environmental exposures and biological susceptibilities to cause and exacerbate reproductive health disparities. These health disparities during pregnancy can also have long-term implications for worse health for generations. So even if the original exposure was removed, we know that the fetus and the germ cells were exposed. And so exposures during an initial pregnancy can cause a trajectory of worse health status. And if the exposures are ongoing, which is currently often the case, the adverse health trajectory is even worsened and exacerbates exposure-related health effects and health disparities. And now returning to chemical exposure specifically, these have been found to be related to many different kinds of health effects and are monitored by the National Health and Nutrition Examination Survey, or NHANES. So they measure chemicals or metabolites in blood and urine in the US population. And Tracey Woodruff and her team conducted a study of chemical exposures in NHANES earlier in 2003, 2004. And they found that the prevalence of detection was very common for multiple chemicals across several classes in pregnant women specifically. So in this subset of about 50 pregnant women, they met with measures of 52 chemicals from 7 classes. All of the women had at least 28 chemicals found in their bodies. So that's, gives you a sense of the scope of exposures during pregnancy. And other work in NHANES has shown exposure to many of these chemicals is higher in low-income women or women of color. However, NHANES only measures about 350 chemicals, which is really the tip of the iceberg compared to more than 40,000 chemicals approved for use in the United States. There are 9.5 trillion pounds of chemicals produced or imported per year, which is equivalent to about 30,000 pounds per person. So there's a massive amount of chemicals being used, and only a few of them are being monitored and studied in the majority of our work. So there's a major gap in our understanding of exposures and their potential health effects in the United States. So I'm going to be talking about some work in the ECHO program, which is a major opportunity to advance understanding of chemical exposures and health disparities during pregnancy and to children. ECHO is a newly launched study that includes over 55,000 children from 71 longitudinal cohorts across the United States. And the sort of turquoise or green dots here represent these 71 cohorts. And there's additional sites doing other work, including intervention studies. Notably, ECHO is about 55% children of color, which makes this a really unique opportunity to not only study chemical exposures across the United States and over time, but also to be able to study and address some of these gaps related to health disparities. So when ECHO began, we also began a chemical exposures working group to try to decide what chemicals should we study, what should the priorities be in terms of ECHO. And so we launched a process to identify novel chemicals. So let's go beyond these 350 chemicals measured in NHANES and identify some more chemicals that are really important based on their exposures and potential toxicity. So I'll describe today this process, which was published in a review paper in Environmental Health Perspectives in 2019. And there's a great deal of information in this paper and the supplemental material. Essentially, our task was to figure out from this huge chemical soup that we know is likely to exist out there, what should we be really studying and trying to learn about? So we conducted a rigorous literature review to prioritize chemicals. And we had to start out with narrowing down from around the thousands of chemicals, how are we going to select some chemicals to really focus in on? So the first step was to find chemicals that were commonly identified in environmental samples or used in consumer products. And that left us with around 900 chemicals. Then we decided to set aside those chemicals that are already measured in NHANES because we know about those. And we also set aside legacy chemicals that are no longer used. Then there were about 760 remaining chemicals. And from those, we chose 155 that could be sort of sorted into 8 priority chemical classes. And we really focused the rest of the review on those. And those chemicals included those in the classes of alternative flame retardants, alternative plasticizers, aromatic amines, environmental phenols, organophosphorus flame retardants, perfluoroalkyl substances, pesticides, and quaternary ammonium compounds. So these are chemicals that are found in a wide variety of consumer products that pregnant women and children are exposed to, including plasticizers and flame retardants that have been noted by others in the presentation. And then for each of these 155 chemicals, we applied a very rigorous literature-based approach to classify them as in 3 categories. The first was whether if they were recommended for evaluation in echo. Those are chemicals we thought we should measure. The second category was deferred pending additional data. For a lot of these chemicals, there's just not that much known. And so we felt like we needed more information before we could recommend either measuring them or not. And then we also classified some chemicals as low priority, where we did not find evidence that they should be something that we should be very, very concerned about. So briefly, to walk you through the process, the first step was to determine whether the chemicals were found in human biospecimens or environmental media. So if we found insufficient information, they were deferred. But if they were monitored but not detected, suggesting that there was low potential for human exposure, we considered that chemical to be low priority for echo. The second was to look at toxicity. If we found insufficient information, the chemical was deferred. If they were low or non-toxic, then it was low priority. And then the third step was to determine whether there was an available biomarker for the chemical. So if we want to monitor it in echo, we need to make sure we can measure it in biosamples. So for those, we either deferred them pending the development of a biomarker, or we recommended, for some, they just weren't well suited for biomonitoring, and you would need to measure them in other ways using a proxy measure. But if they did meet these 3 criteria, then they were considered candidate chemicals for biomonitoring. So I'm just going to walk through one example using the organophosphorus-based flame retardants, or OPFRs. So this list here on the left side are the chemicals that made it to our categorization for priority evaluation. And to note, NHANES does measure chemicals in this class, and so they're important chemicals, but we are looking at chemicals that are not included in the NHANES biomonitoring and whether those should also be evaluated. So the ones that are shaded in orange are the ones that we did end up recommending for biomonitoring and echo. But our literature review went through first for all 155 chemicals to see if we found evidence that there was exposure or they were found in environmental media. So you can see for these OPFRs, most of them were found in biofluids, hair, nails, air, indoor dust, food, and water. So for these, we have strong evidence to suggest that human exposures are likely. The second step was to look at toxicity. And so ECHO is focused on 4 priority children's health outcomes, which are related to perinatal outcomes, neurodevelopment, asthma and other airways related outcomes, and obesity. So we focused our review on endocrine disruption and those related health endpoints. And we looked at in vivo and in vitro toxicity studies, as well as HTP assays and QSAR modeling results. And without going into a lot of detail here, the darker shading basically indicates that there was some evidence or strong evidence of potential toxicity. And you can see for many of these OPFR flame retardants, we found evidence of toxicity related to developmental and reproductive effects, obesity, carcinogenicity, and some neuro outcomes. And then the last step was to determine whether there was an available biomarker. So many of these OPFRs had been measured in either hair, fingernails, urine, or breast milk, suggesting that they would be easily measured in samples from participants in ECHO. So that was our kind of step that we took for each of the 155 chemicals, and it was a massive effort by lots of collaborators to review those literally like over 1,000 studies. And of the 155 chemicals we reviewed, we found that 36 were high priority for biomonitoring and ECHO, that they had high prevalence of exposure or predicted exposure, that they appeared to have toxic properties, and a biomarker was available. We deferred 108 of the chemicals, and a lot of the times that was due to just insufficient information. And for some of the chemicals, it was because we did not have a biomarker yet. And then we found that 11 of the 155 were low priority, in that we didn't really find high evidence of exposure or toxicity. So now that we have this priority list of chemicals that are sort of underneath the iceberg that we think are toxic and have highly prevalent exposures, what are we going to do with this information? So we decided to take another series of steps working towards a large epidemiological study of these chemicals in relation to children's health. So the first step is now that we have a set of chemicals we want to measure, we need to develop an assay. And so we partnered with Cannon's Wadsworth Laboratory and he developed a multi-class assay that measures 102 biomarkers in urine. So these 102 chemicals include the ones that were recommended through our literature searching, as well as some that were deferred because they needed a biomarker. So Cannon's team worked on developing a biomarker for those. And we also included other chemicals that not in the original list, but could easily be added to this assay to make it more useful. And these chemicals were those that had similar structures or sources to the chemicals that we had already identified. So this is a really excellent way to use resources wisely and measure a large number of chemicals that are really of concern for reproductive and developmental health. Now that we have the assay, the next step was to conduct a pilot study to measure these novel chemicals in urine from pregnant women. And this is what we're doing right now. And Tracey Woodruff is co-leading this with me. We're doing a pilot study to measure chemicals in 175 pregnant women across 9 echo cohorts. So we've selected cohorts across the United States and including Puerto Rico in order to capture geographic and temporal and socio-demographic diversity and begin to determine the prevalence of these exposures and some of the predictors. Notably, this sample reflects the diverse race and ethnicity distribution of the full echo cohort with about 50% of participants of color, so we can assess not only exposures, but also whether there are potential disparities. And this pilot study can inform future hypotheses and targeted studies to assess these exposures. So right now we have some initial information from the pilot study that these chemical exposures are likely very prevalent, and we have begun to conduct the next step of the study, which is to measure these novel chemicals in a diverse sample of more than 7,500 women from 22 echo cohorts and assess associations with birth outcomes. So this will be the largest study of chemical exposures in the United States, specifically addressing these novel chemicals that have not been measured before. And we're starting with birth outcomes, but then once we have a data platform where we've measured these chemicals in a large number of women, can perform multiple future studies using the ECHO data set, focusing on other priority health outcomes such as obesity, asthma, and neurodevelopment. And with the diverse socio-demographics of the ECHO study, we can also address some of the important questions with respect to health disparities and exposure disparities to these novel chemical exposures. So our goal ultimately is to fill some key knowledge gaps regarding these contemporary chemicals in order to influence evidence-based decision-making. Our study will be the first of exposures or health effects for the majority of these chemicals. But if our hypotheses are confirmed, then these exposures can really be reduced through a variety of different actions that can be taken, on the individual level, the policy level, and consumer advocacy. Importantly, other studies of chemical exposures during pregnancy have found, important health effects and motivated policy changes have resulted in lower exposures. So hopefully we can identify those chemicals that are of the highest concern and begin to protect children's health. And we think, if we can avoid exposures during pregnancy, it's not just that pregnancy that will be having better health, but for generations. We know that it could have long-term implications. So with that, I just want to thank a few of the collaborators. This is a really large and amazing team of investigators in ECHO. And I just want to note Tracey Woodruff, who is co-leading the studies, Ido Palazzari and Debbie Bennett, who led the review paper, and Cannon's lab, who's done so much to be able to quantify the chemicals. Thanks.
I'm now talking. Thank you. That was really excellent. I think, Hannah, right, our next activity here will be to have a few questions that came up during the discussion, and then we'll take some questions from the panelists, from the people who are very actively participating in the chat online. And I was really struck by the discussion about that you pointed out about how while we have some knowledge, we have these key kind of data gap areas, whether it's in chemical monitoring or monitoring around testing protocols or measures of health, reproductive health in the population. And I wondered if you each would identify two opportunities, or if you could wish two things that the government would measure or implement in order to improve our understanding of this challenge, what would it be? I want to hear, because I'm thinking, maybe next week we'll have a good opportunity for recommending those. But you have to go off mute in order to answer.
So Tracey, you want to know specific chemicals or regulatory?
Well, I'll just go to you, Shanna, because you mentioned we don't have good animal or in vitro testing for these endpoints. What would be two specific areas that you think would help us better identify these chemicals that can affect male reproductive health?
Okay, so the way as you know, Tracey, and many people know, that the way chemicals are tested, they are not tested the way people are exposed, right? So they're not tested in mixtures, which we're exposed to, and they're not tested at low doses that people are exposed to. So we don't really have protection even for the few chemicals that have been tested. So we have a dearth of chemicals being tested, and we have them not tested in protocols that protect people or the environment. So those are my two things, I guess. Low doses.
Germaine?
Yeah.
I was going to ask about the mixtures. That's a good point. Just Germaine?
Yeah, so I'm going to focus a little bit more on the health outcomes, if that's okay. So we do
yes, definitely
national screening and have surveillance systems up certainly for birth defects in many, but not all states. We do have it for cancer nationally and a few things like that. I think we really have to make fecundity related impairments, urologic and gynecologic disorders important too. And not just the testes or ovarian cancer, but things like endometriosis, polycystic ovarian, all those sorts of things that really take a lot of clinical time and cost a lot to manage, over the course of one's lifetime. So why not think about mechanisms if we really believe that these could be in the pathway to continual onset of diseases or things like that? Why not try to get out in front of it and to get an answer? It's very hard to even know, has there been an increase in endometriosis or fibroids over time, right? I mean, it's very hard to really know that, except in societies that measure it better. But for earlier on, things like semen quality, if it's true that poor semen quality means you're going to die sooner, right? Then should, physicians, pediatricians, and early internists or something, should semen quality be a part of the physical assessment? Would people want to know? The problem with something like time to pregnancy and maybe even early pregnancy loss with women now being the gold standard, knowing, doing a test, knowing they're pregnant, then knowing that they had the loss. How do we do surveillance in people's homes? So somehow I think we have to change the thinking about human fecundity, that it's maybe the canary in the cave, right? It's the early sense of whether or not you're going to have related things, that will be compromising your health. So I don't know exactly how to get into people's homes where some of these hidden endpoints are happening without some sort of culture where it's okay to talk about sex, basically sex that gives you those outcomes. And I think our country, relative to a lot of others, we're uncomfortable with sex. You can tell I'm not a fit
I'm just like, that is a really great comment. And I'm like, we're not going to pursue that at this webinar. But Germaine, maybe Che will have you back again to talk about that. Jessie, did you have a thought?
Yeah, I mean, I completely agree. I think another thing we spend a lot of time doing is studies to try to identify the sources of chemicals, or what are the things that are causing exposure. So with a limited labeling requirement and not knowing necessarily where these come from, that's another big gap.
So I'd just like to add one thing, which is that we haven't, any of us, talked about the link between these chemicals and climate change. And I can't leave without mentioning.
Well, go ahead, Shanna, please talk about that link.
What?
I think I know what you're going to say, but I want to hear what you have to say.
So chemicals in plastics are made from petroleum byproducts. Many other chemicals are made from petroleum byproducts. The ascendancy of the petrochemical industry and the plastics industry has gone hand in hand and has correlated with the declines of 1% per year in many areas of reproductive function that we've been seeing. This is a whole picture, and it has to include climate change.
Thank you. You said it excellently. I would also just wanted to remark on, we focused a lot on, or you panelists all talked about primarily about human data, but I just wanted to lift up this idea of the conservancy across the animal species. And I think one excellent part of, Shanna, of your work is looking at the toxicology literature which had been around for 100 years using this marker of male reproductive health. Very clever, but I think we have more, opportunities within related fields of ecology as well as animal health to advance this. So, Hannah, I want to check with you about timing, because I know we're perhaps we should turn it to the questions.
Yeah, we can go over about 5 to 10 minutes if the speakers are okay. Do you guys have time to go over a bit?
Yep.
Okay, thank you for your time. Yeah, so if you wanted to continue the conversation for a few more minutes and then jump into questions, that'd be great.
The only other thing I wanted to just get you guys alluded to this about the cumulative exposures element of this whole paradigm. There's two things. One is, a lot of the studies are on just individual level exposures, and we know people are exposed to many different chemicals. So is there a clever way we don't have to repeat all these studies, but could think about how to integrate that? And I also wanted to just highlight this idea about using these upstream markers, perturbations. Germaine, you talked about, both of you guys talked about the dysgenesis syndrome. You had different labels on top of it, but it kind of aligns with some work that's been going on around either key characteristics or looking at these pathways so that we can better mark those, because the downstream consequences are many, and sometimes too diffused for us to really combine in that thinking about them more upstream. I'd like to hear both your comments on both those items.
Well, I do think that amongst the scientific communities, there's a more open approach and a willingness to be able to merge cohorts, maybe a different, you might have a pregnancy cohort and a children's cohort, an adolescent cohort, and think about ways such as the ECHO, where we can bring together existing cohorts and not have to wait another 50 years to look at a new cohort, whatnot. And, to be honest, there was a little resistance to that thinking, right? Because of the heterogeneity and a whole host of other issues. But I do think coming together to leverage data that's out there, even if it's not the same data from, all the data from one cohort, is a major paradigm shift. And I think this existing generation, the ones to come, will figure out better ways to get answers quickly. So I'm supportive of just trying to, work on the method side to help advance those questions, because we can't wait another 100 years. That's for sure. It's just too
I really like that idea of using the existing, like doing a better job of mining what we already know, because we actually don't do a very good job of that. And that idea of being willing to look across, you're talking about epi cohorts, but also the animal data to do a better job of aligning it up so that we can just take actions quicker. Sorry, the other panelists want to speak about this?
So I'd just like to go to the question of, you know, looking at mixtures. There are excellent methods being developed, and maybe Jessie would talk a little bit about that because they were using those in ECHO, and in other studies, of course. But I'd like to add that because we as epidemiologists don't often go to animal studies and don't take advantage of the rich information that we can get from those, which are relevant to us, because otherwise why would we be studying them after all? And you can expose an animal to a mixture of chemicals and get very good information on mixtures, much of which is relevant to humans. And so I think there has to be much more interplay between the animal studies and the human studies, because we don't have time. We don't have time to wait to study all of these things in humans, you know, in 2 at a time and 3 at a time. I think we need to ramp up the interdependence of the human and the animal studies.
True, and we are animals, so just
yeah, right.
people.
I also think there's kind of another way that we could do it, and this is something that we have partnered with in our endometriosis study, the ENDO study, and that is epidemiology teams and clinical teams. You collect these wonderful biospecimens, and then we observe these associations, this chemical and endometriosis risk or something of that nature. This is an opportunity for a basic scientist to get involved, to try to do mechanistic work informed by the epi, to help us understand what those associations mean. And to me, that has been so rewarding in my career, those, you know, because you have good phenotypes, you've got good exposure data, sometimes the things that you may not have, you know, in a lab in a big way. So I think helping us understand the associations with good mechanistic work is a really fruitful opportunity as well.
Jessie?
I just think some of the best new biostatistical methods for looking at mixtures are those that are pulling from the animal studies, and you can use, you know, there's some new approaches that are using, you know, quantitative chemical toxicity data to try to kind of merge that in with the exposure data and help elucidate the pathways. And I think those approaches will really help us understand and leverage the data.
Great. Hannah, thank you guys all for your really insightful comments. Hannah, is it time for us to turn to the Q&A?
That'd be great. Yeah, let's get some questions answered here.
I don't know how you're going to choose. There's so many here. I'm going to let you. I just wanted to briefly pinpoint the discussion about the quats, because I know we said we weren't going to measure it in echo, but I do know that the state of California has prioritized those for biomonitoring in their statewide biomonitoring program. So there could be methods development going on in the state. So I just wanted to highlight that that's still ongoing. And maybe when you look at Jessie's slides later, you can pick the chemical that's my favorite that we're going to be looking at. So anyway, Hannah?
Yes, we have Karen here too. She's been going through the Q&A. And Karen, do you want to jump in?
Sure. Let's see. Let's see. Someone asked, it's been 20 years, Shanna, since phthalate syndrome has been identified. Have there been any multi-generational follow-up with children of original infant participants?
Not in humans, unfortunately. And this is something that really needs to be done. For our study from 2000, you know, those kids are now 21. And, you know, so the problem is that it's very hard to find them. It's very hard to track them. So I think there would have to be a real commitment on the part of NIH to do that. It would be costly and valuable, incredibly valuable. Imagine we have all that information on the parents. We have, actually in that study, we have information on the father's sperm count even, you know, and the mother's chemicals and the mother's health. And then you have, you know, the potential to get all that information in the children would be huge. But it's going to be expensive. And I don't know. I don't know if anybody else has another response to that.
Yeah, Shanna, can you talk about some of the health endpoints that you talked about measuring? And specifically, we had someone ask about, does the decrease in sperm counts relate to sperm motility? And, what can we infer about, fertility from decrease in sperm counts?
Right. So of course, fertility is not sperm count. It takes two to tango. There's lots of other parameters that go into fertility, infertility. However, the rate at which fertility has been declining worldwide is actually also about 1% per year. There's wonderful data from the World Bank. You can look at an interactive map. You can put in the years, the countries, and you can see that, all worldwide. So the other endpoints that have been declining at the same rate, there's miscarriages have been increasing at about approximately that rate. Genital defects have been increasing. Testicular cancer has been increasing. So there's a total picture that is captured, if you want, I call it the 1% effect, that these things are changing at approximately the same rate, and it's in males and females. And as to the question of what about motility and morphology, other sperm measures, we didn't look at those, and no meta-analysis has looked at those because those methods have changed over time, while counting sperm has not changed over time. And that makes that very difficult to study longitudinally. However, in individual studies, we see them correlated. So men that have low sperm count tend to have lower motility, lower morphology. So they go together in and it's probably, if I had to predict, I would say there has been a decrease over time, but we haven't done those studies.
Okay, one last question for you. Someone was wondering if the AGD can also be a measure that can be used in human females as an outcome related to phthalates as well.
I'm so glad you asked that question, and it's definitely a measure in females, but not related to phthalates, because phthalates affect androgens, and females do not need androgen for the development of their AGD. But we do see, in our study, we saw that women with PCOS actually had children, girls, who were born with a longer AGD. Well, women with PCOS have higher testosterone. And so that is higher testosterone in the mother is reflected in longer AGD in the females. Lower testosterone in the mother is reflected in shorter AGD in the male. So that's the counterpart of that.
Great. We had somebody ask, are there any major differences in exposure rates based on race and ethnicity that can point to the relationship to environmental injustice? That's a general question, and any of the speakers can address that.
Well, I can say there are certain, I'm thinking about some of the persistent endocrine disrupting chemicals, some metals that have been associated with different population, racial, ethnic groups, sometimes on the basis of where they live and maybe contamination in their area. And we've heard about a lot of those locations, or else in terms of diet that could be associated. In the fecundity work, unfortunately, so much of the work doesn't have a lot of diversity in terms of the study cohorts. And so it gives us a very incomplete understanding of how those differences might be operating for some of the non-persistent chemicals that might have more of a prevalent exposure for all of us. We may not see the differences, we may. I know Shanna has mentioned in one of her studies she has. In some of my work, we haven't seen persistent differences between racial ethnic groups, again, usually with maybe 20% of the cohort comprising non-white population. So I'm always hesitant to say that it isn't there. So maybe I'll defer to Shanna, and she can talk a little bit about what she's observed in some of her work.
Thank you, Germaine. I think the study, you were referring to my comment that in our study, we found higher BPA exposure in African-American women in our Rochester cohort, and that is true. But there is a pretty substantial literature, and maybe Tracey, could you talk about Amy Zoda's work on this?
Well, I would say there's a couple areas. First of all, the biomonitoring work, we've definitely, and Jessie can talk about this. So we've seen that there's certainly some of these persistent compounds. When you look at broader populations, you see higher exposures in certain racial and ethnic groups. So work that's done has shown that PBDEs are higher in African Americans, for example, in California, or they're higher in low-income communities and communities of color. And this I want to just extend because we've talked about biomonitoring, but we haven't really talked about sources of exposure. So work has been done to look at like presence of chemicals in dust. So Ami and Rachel Morello-Frosch have done some work looking at communities in California, comparing low-income communities of color to higher income, more white communities, and finding that the level of chemicals in the low-income communities of color is higher. And if you start to map sources of exposure, so you look at where people live and you aggregate sources of exposure, like from freeways and highways, or chemical plants, or refineries, that you tend to have this more concentration of these sources of exposure. And you can see this, California has a CalEnviroscreen, which maps these sources of exposure and how it overlays with other stressors that increase health outcomes like poverty, segregation, already pre-existing conditions. And what we're seeing is this confluence of these risk factors in more low-income communities, communities of color. So like you see the Richmond or East Oakland, areas in the Central Valley, San Bernardino. So I think it's like you have to look at the whole picture. That's why sometimes when you look at an individual cohort or specific things, you only see one element. And I think Germaine's comment about expanding this across to ECHO, where we're going to link in multiple cohorts, we'll have a much better opportunity to look at this, because, I don't know, Jessie, maybe you want to talk about some of the cohorts specifically recruit Black women, so we'll be able to like enrich our understanding of that group for these as we look at this biomonitoring. Did you want to say more about that, Jessie?
Oh, just, and there's differences based on occupation that have been found. And there's also certain personal care products that are used in different populations can carry high burden of chemicals. So there's lots of, I think, following the source is really important.
I just have a question for Jessie. Do you think ECHO would consider a study that's not outcome-based, but that would focus on parameters that predict exposure levels?
Yes.
And we've been chatting about that, haven't we?
We have, and I've been also trying to recruit someone to work on specifically looking at health disparities in chemical exposure. So I have a couple of people I'm talking to about that as well. I think, ECHO is a perfect place for that, especially among pregnant women.
So we've actually been paying attention to the questions we're asking the participants about sources of exposure, using some of the studies like Ami's done and other people, so that we can look at some of these things like packaged food and where do you live and the kind of products you have in your house. So we'll have the data to be able to look at that potential about people saying I'm using certain things or I'm living in a certain area, and then we'll have the biomonitoring data, so that'll help us. Because one thing I want to also note about ECHO is it's really focused on solution-oriented research. So how can we do research that will solve a child health problem? And I think that's why, our expanding the biomonitoring pie is so important, because what biomonitoring is, of course, downstream of where we want to be. It does also help us think more broadly about all these other chemicals that are out there and measuring them. And again, there will be some many interesting results from that, I know.
Okay, great. This webinar in this series is about reproductive health, but somebody asked, can somebody comment about, how much crossover there is on these environmental exposures, EDCs, and other health systems, outcomes like the immune system, etc.
So I'll just say that we're studying our children in our TIDE study, which by the way is in two ECHOs, two ECHO centers, they're now almost 10, and we've been following we have their reproductive development, and now we're looking at their neurodevelopment as they get older. And we're seeing that a lot of the same chemicals that are affecting their reproductive development are also affecting their neurodevelopment. So I think when you interfere with basic hormones in early pregnancy, you're going to mess up all the systems. I think that's my prediction. And, you know, I wouldn't be surprised if any link to a childhood later outcome being linked to those same chemicals that have affected reproduction. The reproduction is like if you will, the tip of the iceberg.
Okay, great. One final question, and we're over time. And any of you guys, maybe all of you guys can respond. Despite the mounting evidence and data, there's so much doubt among the general public. How do you respond to people who either don't believe in this, and what can we do to get protective regulations?
I guess I would say that communicating risk and uncertainty is tough and it's challenging, but I do think there are disciplines that are really committed to trying to get the messaging correct. And at least with our studies, even though we were measuring all these chemicals, we gave back the results to the study participants, I believe quite strongly in that, even when we don't know if it's a safe level, what does it mean for them? And we haven't done things like talk to your doctor, because we know that just is a punt. Most of the times the average internist wouldn't know what the chemicals are or what to do with it. So we try to provide some sort of context, and if there are metrics that are out there for health alerts, something like lead or other sorts of things, we try to provide that context. So I think it's a challenge because we make people feel so powerless, that these chemicals are all around, they have them in their body, and they can't do much about them. And as a result, I think people turn away and then don't pay attention. So I think we have to be careful with our own messaging in terms of what does it mean that you're going to be exposed, and be focusing, as many investigators out there and authors are, how to minimize your exposure. You know, it's not a done deal that you're going to have exposures, but here are ways we can empower people. And I know that Shane and the other organizations, many of them logos I see in front of me, have really good web pages where you can have down-to-earth information and still feel somewhat empowered to help protect your own health.
Yeah, I just want to follow up on that. I agree that first of all, I think having this type of webinar series and venues to talk about the science is really important. And it was really amazing to hear the talks from people who've been in the field a long time, to appreciate that we need to do a better job about moving that science into an action that will reduce exposures. I will say, though, that, and Shanna didn't really talk too much about this, or Germaine, but their work was very instrumental in laws that have been passed in California and the United States and in Europe to ban certain types of phthalates in certain types of products. That's the kind of, and it has led to reductions in some phthalates. So there's been substitutes of other phthalates. So I think that the public and the environmental health community and some work that we're doing in the clinical community are engaged and understand that this is a problem, that have been making efforts into this. But it is true, we need to be more comprehensive and preventative in the policy approaches. I would say that, you know, there's a lot of opportunity within the federal agencies to do this. The Consumer Product Safety Commission, people have been working with them to have them ban products, FDA. And then the Environmental Protection Agency is really an important focus for activity because they administer the Toxic Substances Control Act law, which really governs chemicals, allowing chemicals and to what extent onto the marketplace. And I think that one of the challenges that we have before us is that they have not been using the best science to implement that law that was updated in 2016. So it's really behooves being engaged with groups to advocate on behalf of using the best science. And I know that that's something that we're doing. And then of course, because the government controls the exposures we have to to these industrial chemicals, everyone in the United States has a say in the government, and that's by voting. So I encourage everyone to vote. I think people know there's an election next week, right? And as a member of the UCSF community, we are encouraging everyone to vote in the election to let everybody know your opinion about who should be making decisions in relation to everything on our health.
Yes, thank you so much, Tracey. And thank you to all of our speakers today. Hannah, we'll hand it back over to you.
Yeah, thank you so much, Tracey and Shanna and Germaine and Jessie. We are approaching the end of the webinar today. The video recording will be available on CHE's website soon, and tomorrow you'll receive an email containing the link to the video. The next CHE PRHE partnership webinar in this series will take place November 18th and is titled Environmental Reproductive Justice, Racial Disparities in Environmental Pollution and Chemical Exposures. You can find details on our website at healthandenvironment.org. If you are new to CHE and would like to stay updated about upcoming events or more, please sign up to receive our newsletter by selecting the Join Us tab at the top of any page on our website at healthandenvironment.org. Additionally, if you appreciate these CHE partnership webinars bringing you the latest environmental health research for free, we encourage you to support CHE's ongoing work by making a tax-deductible donation via our secure website. Again, our website is healthandenvironment.org. With that, I would like to thank our speakers, Dr. Shanna Swan, Germaine Buckley, and Jessie Buckley for taking time to present today, and to you, Dr. Tracey Woodruff, for your excellent moderation. Thank you so much for joining us. We are wishing all of you much health and wellness. Have a great day.